thank you for everyone to attending the presentation. So I'm very glad to share what I have done on the topic of the application of the biological control based on the study of fungi, insect, and houseplant interaction. So now I'm currently working in the Louisiana, but I come from north of China. The Changchun capital of Jilin province, very close to Russia and Mongolia, this area. So my hometown, where the environment similar with American, especially in Michigan. In summer, there's a lot of lakes and rivers, but in winter, the temperature can get into minus 35 and 46 degrees. Everything have be frozen. So when I moved to Michigan, I feel Michigan is so warmer. So I like here. And then I worked in the, I have visited as a witness scholar working in USAID for several years, which is located in the campus of Iowa State University in Ames. And also working in the Pacific Biological Research Center located in the campus of the University of Hawaii, and then moved to University of Michigan for 1 year in Ann Arbor. Now I'm working in the Louisiana State University in Baton Rouge. So I have two ecosystem I'm working on. So first is in agro ecosystem, second is in street system. In street system, I'm concentrating on emission plants, which have been caused serious damage in American stream steroid, but environment. But in China, in the native area, I found the kind of biological agent named kind of vivos. This kind of biological agent can be the host of the biological agent can be limited by kind of fungi because the larva can live off this kind of fungi. I can control the fungi, leave the vivos host range down to only one kind of the plants. Japanese not weed. If somebody interested in Japanese weed, you can contact me. I know all the biological character of the vivos. Two-thirds of my job working in the agricultural system, studying about the pests in the fungi and the crops. First of all, I'm very concentrated on the biotechnology using population genetics, like to answer, get some basic research for the biological control. Another 1/3 of my job working on gene-fied, genetically modified crops, do the screening assessment. The funding is good, so I like the work this. But today I'm going to give this part of the content. This outlines that the green part is that genetically modified crops testing, assessment, and that's the kind of invention, biological control weed, and also use metabolomic analysis, discovering special gene, and also have development kind of software named as gene ads to analyze GPS population genetic data. So I will not give the introduction of them, maybe later. So today I'm giving the introduction of the entomology, fungi, insect, and house plant, and also do some population genetic analysis. The first part I'm going to give the introduction is based on the telomorph and entophyte of Bassiana. Entomorph means sixth generation of the Bassiana as that kind of biological control agent named Bassiana is kind of fungi, and entophyte means like establish a symbiont with the corn. The purpose I want to do for the implication is to produce organic food. Organic food is growing worldwide, but the Eastern Asian population decreased sharply. It's very hard for people to work in field. So people have a high demand for organic food. So population decrease, food requirement increase. Now, a lot of people want to do this kind of research. One of the issue is for the organic food is the price, the cost, how to decrease the cost. The first one is that use the fungi. Fungi is very easy to produce and use the drones. So each drone, you can lift it up and go and come back just in 5 minutes. One drone can cover 1 hectare in 5 minutes. So each time you can get 5 hectares, this kind of things. The cost is very low, just about $10 1 hectare in 5 minutes. So very efficient and decrease the cost sharply. I will give the introduction of the Biocyana. So Biocyana can cause the pests into muscardine. The muscardine can be used by low control with pests, but another muscardine can to be a pharmaceutical compound. But not many people study the telomorph, the sixth generation of them. Its scientific name is cordyceps Biocyana. The Biocyana can penetrate the epicuticle, the supersurface of the skin, and penetrate the epidermis and into the chemicals, produce the loss of the spots and cause pest death. That's the photos about the apophysis in the surface and penetrate the epicuticle. The traditional produce in the industry in China include the 3 steps. The first step, the research scientists have collected the strains in field year by year, and then do the single spot isolation. Then we get the progeny strains. The progeny strains is stock the strains. When we want to get some experimental and reproduction, we can get this from the stocks. But this process have some challenges that it have changed. It can be get excessive methylene growth. So we'll get very little spots in the Canadians. So and diminish their toxic pests. But there's a traditional method to overcome this issues. It's screen the well the strains. It means collect the strains year by year. Another method use some chemicals like the UV lights, but it still can get only some mutation side, cannot get whole genomic exchange. Another advanced technique includes genetic modification and protoplast fusion. This can cause some low side exchange, but very limited number. The question is that evolutionary speed is very slow because cloning living cycle, so no allelic exchange. Another question is strain diversity and from mutation side and the natural connection, so cannot get enough mutation side. The third question is telomorph is hard to find in nature. So far, there's only one strain was found in the border of China. The second is limited worldwide results. So you collect some locations year by year. So the source is a limitation. Even the gene modified strains it's not safe. So it will cost about 10 years or 15 years to get the permit to apply in field. I figure out 2 methods to solve the issues. The first one is using the telomorph, 6th generation of the Biocyana. So this kind of Biocyana belongs to the mating system. In Eskimosites have 2 kinds of mating system, heteroxylum and homoxylum. Heteroxylum means different strains containing different mating type gene. The homoxylum means one hyphae containing 2 kinds of mating type gene. They can self-fertilize. Biocyana belongs to the heteroxylums. Both of this kind of the mating type system can get sexual reproduction and formation occult spots. I have do some screen about the strains which stored in my lab. I get only 4 strains which containing 2 kinds of mating type gene. All of the strains contain only 1 mating type gene, MAT1-1. So I assume that potentially MAT1-1 gene have played very important role for the evolution. So I do that kind of things. Belongs to the heteroxylum. Each strain contains different mating type gene. I do a lot of single spot isolation. I get different strains, substrains from original strains, which containing different mating type gene. So I do the biological characters investigation. All the strains containing MAT1-2 gene grow faster than the MAT1-1 gene, but they get very little less quantity comparing to MAT1-1 gene. how to capture it. So how to get the telomorph. I have successfully captured the telomorph on the pupae and also on the rice. Use about 1.5 months, we can get the sporophores come out from the pupae, and use another 15 days, they change the color from white to the golden, and then use another 15 days, eschers will establish in the top of the sporophores. I cut it sporophores stick on the cap of the petri dish. You are about 17 days later, the new Canadians and the spores pop out of the eschers. We can get a lot of colonies. So each colony is which containing a novel genetic background. It means that we have one very limited results of the strains. I can become it. The results infinite. So I'm thinking about another thing that even they can the plant can absorb the Bacillus spores, and the spores can walk through all the body of the plant. It's called established symbiont. This kind of things called introphyte. So earlier any development things happens, I don't know, so I designed this kind of treatment, use M81- gene, M82- gene, and mix the treatment to do the assessment. So the treatment one, M81- gene, M81- gene, and mix the treatment, I still get the single mating type gene strains from the two treatment, and the mixed treatment, I get the two kinds of the strains. I assume just as all the strains containing M81- gene, I assume that they play a very important role in the evolution. So I do the sequencing for the M81- gene. I got tons of the mutation site, M81- gene, but I get 0 mutation site from 81- gene. So I think that I will concentrate on to studying the M81- gene. So this is a mutation side as include transversions and transitions. Earlier some character changing or some development things happens comparing to before absorbing application of the Bacillus. So we found that after recollection, I got a lot of the colonies. So all of the growth factor a little higher than the before pooling, but the mixed two mating type gene get a higher mortalities. So it means that in the future, if we use to apply in field, we should use the mixed treatment better than the effect of the only one strain with containing only one mating type gene. The conclusion, we think that telomorph can reinforce its evolution in nature by artificial methods like this, and M81 gene played very important role for the evolution of it. Okay, so another application of the new strains. So I get the new strains as our new product. How to apply in field? It's based on the co-evolution study. Is that first of all, give a short introduction of the target pest. The target pest is that named the Ostrinia nubilalis. In the first star and the sixth star is the only occurrence in the column area, but after the third star, it will born into the stalk of the corn. It's the female and female. If the first star penetrate the rows of the leaves, when the leaves spread, we can get the holes in those. It means the first generation impact. The second generation, the mouse will lay eggs and the leaves very near to the ear. So the larva will attack the top of the ears. So it's for the second generation attack. Usually people apply Bacillus in field use drones. And also they collecting some stalks in piles and use the granule, put them into the inner piles of the stalks, like a kind of steam. So overwintering larvae will die from infection, get infested by the Bacillina, the traditional. But the question is that it's very hard to get the optional time because the first star and the second star exposure outside only not more than 2 weeks. The second challenging is the control Bacillina, which one have born into the stalks. So how to decrease the cost? Before we doing that, we should have to do some investigation. Usually people collecting egg masses in field to get the occurring pattern and the peaks number. The first generation, only one occurrence. The second generation and the next generation of the Asian corn borer. But the option time is not on the peak, it's 1 week before the peak. So it's very hard for the technician to get the optional time. With the global warming, the generation have changed. This is gene preference map in about 35 years ago. So one generation error, second generation error, but now it's have 2.5 generation. So this occurring pattern is not stable, it's changing with the years. So I use my new strains to control it, to control the pest, use the optional time. I have set 2 controls. The first control is first generation, second generation, and the 2 treatment for the 2 generation. So my strains can get still decrease the live feeding and bore holes. It means that the new strains can get a very good control effects. So how to decrease the cost? I just developing a method like a granule method. I make a new vector which is made from the dry stalks. This kind of vector is very different from traditional granules vectors. Is that it can be loaded into the joints, into the columnar area. This area can protect the Bacillina from sunshine and keep the moisture. So this kind of the vector can make the Bacillina produce spores during the whole summer. So it's kind of keep controlling effect. Another method I will use it. It can mix it with the trichogramma. Trichogramma is this kind of parasite wasp. So I put the Bacillina spores into the gears of the trichogramma. So when the wasp will find the egg mass and it can brush the spores on the surface of the egg mass, so decrease the cost. Usually, one optional time have to be applied 2 times, but using this method, it can narrow down use only 1 time, so decrease the cost too. Another application for the entomopathogenic application, based on the for the one who have born into the stocks. The question is that we should first of all we should answer this question. So whether the Bacillina can penetrate the digestion system like the middle gust. The second is how to apply it. We cannot pull all the suppression one by one. So are there any effect for the second generation? So I do some bio-eyes. So I have to mix the Bacillina spores with the artificial cell and inoculate the first star or the levels in corn borer. About a little more than 1 week, I got a lot of muscardine. So I do the transmission electron microscopes. The picture in first row is presented which one on the skin and the epicuticle of the levels, and the second one is presented in the mid guts. It means you know that about 1.5 days the spores start to development, and after about 2 days it can break through the mid guts. It means that it can cause the target dead. It can penetrate the mid guts. Okay. Oh, the dipping and dipping, what's it? This one?
Like the top panel, it's like 12 hours, 24, 36 after dipping.
Okay, it's the first picture is not so I mix the spores with the artificial diet. So the larva feed on the artificial diet. So the Swiss will get the spores of the Bassiana and also eat some spores in their meat gut. So the first top is the time similar with the time, the effect of the in the meat gut similar with which one on the top of the surface. So the time is similar, have similar effect. So on the top of the surface, get the spores can still development and penetrate the skin of the larva about first level, first instar level. So if the second one is in the mid gut, the spores can development can to develop and penetrate the mid gut, cause the larval death. So two kinds of the method to cause the larval death. The first instar larval. You're welcome. So how to apply it? I found that in the dry area and half dry area, the deep system will be used very popular. So I set a tank, just put the suspension containing the spores into the tank in right tank. And also I want to keep all of the plants get enough egg mass. So I infest the egg mass in the column area to do its assessment. The interfied application in biocontrol pests, I use always use two kinds of spores. The spores which culture from the solid culture medium. The second is from liquid culture medium. The two kinds of spores cause different control effects. Yeah, I'm using my own strains, my own new strains, which cultured by the telomorph. We can get that the mixed treatment can get a very lower live feeding rate. So in the it tell us that in the future we should use the mixed treatment. The mixed treatment is that two kinds of the spores, the Eros Canadian and blastospores. So I mixed them to put it into the tank. This is the result of the assessment that the CO1 and the control force and the control site means the first generation control and second generation control. So the first generation application and the second generation application still can get a good control effect. It still can decrease the borehole and the tunnel length. And also use the first two figures present the granular method. Also use the interfied suspension into the tank, still can get good control effects to decrease the borehole and the tunnel holes. It's very interesting that they pull a lot of the so much suspension of the Bassiana. Are there some effect on the plant? I found that 1 month, 2 months, after 3 months, it can increase the average length of the corn. It also can increase the upground weight and underground weight. It means that deep every day is with the suspension Bassiana, it can accelerate the growth of the plant. So I also do some promotions and demonstrating my two techniques. I base my own base to do exhibition and also do some organize some course and lessons to treat local scientific technicians and some farmers. Also, the local TV station reported these two techniques. Another part I want to give the introduction that the population genetics. Population genetics is so all the control should based on the population genetics. We should know the occurrence pattern and what they are. This is this part what I have done. Asian the corn borer have two sibling species, Asian corn borer and European corn borer. Asian corn borer spread from eastern China to the Australia east. The European corn borer original from Europe. In 1990s, it had made invention into America. In the Eastern Central Asia, like Xinjiang province, west of China, this area about 40 years ago, only European corn borer occurrence here. But recently, about 7 years, I found Asian corn borer moved here. The two kinds of sibling species have been separate for many years because of deserts and mountains. But now, what happened when they meet again after millions years? Usually, it's very hard for us to separate by morphosis, and outside very difficult to separate them. Usually, we separate them using the pheromones. So I've collected about 11 locations in that area. So Xinjiang province, they have been separated by the high mountain and desert into 3 parts. The north part, the south part, I see them as the allopatric area. In the same patric area, like have 5 counties. The color red present the European corn borer, the green present the Asian corn borer. So the mixed pair is a mixed occurrence. So before you apply it, you should know the target. They have similar niche, but not very much similar, not much same. They have different house preference, but they all attack the corn in field. So I'll extract the mitochondrial DNA. I found that the haplotype can separate the Asian corn borer from the European corn borer. Mitochondrial DNA, I use the CO1 gene and the CO2 gene, and then connect the 2 genes together, and then do the evolution tree. I also do some gene flow, 5 FST. The first 5 column present the same packet area European corn borer. We got the 5 FST. The 5 FST have a high value. It means that have a high gene flow among all the populations. But the Asian corn borer in same packet, they also have the high gene flows. It presents that they can separate Asian corn borer to European corn borer greatly, efficiently. The earlier, another question I want to answer is that are there any introgression happens between the 2 species? Because there are no reports they can hybrid. So I'll do some resequencing, whole genome resequencing for the 2 species, and screened about more than 200 SMP loci. SMP is single nucleotide polymorphosis. I have used about 200 SMP loci. I'll do the structure analysis, and the decay was maximized at 2. So the first 5 columns present the individuals occurrence in the same pattern area. It seems that in corn borer, some individuals share some genes with the Asian corn borer. It means that they can hybrid in nature. It's the first time to report that they can hybrid individual, they can hybrid in nature. So I also do some special clustering analysis. XY means Xingyuan County, match with this one, Xingyuan County. The individual match well. The color means that they have a high gene flow between the 2 sibling species. I also do the PC analysis. I have matched all the individual potentially hybrids and match all of the individual well. So it's another proof that evidence that they can hybrid in nature. So another phenomenon we want to do investigation about voltinism. Voltinism means that how many generation occurrence in 1 year. Asian corn borer is very have genetically voltinism, include univolting and biovolting. Univolting means that the individuals occurring only 1 year, 1 generation, despite how good the environment it is. But the multiple voltism or biovoltism individual, they depend on the environmental condition. They have different generation in 1 year. I collected the sample in the Jilin province, Eastern area, one generation error, second generation error, and mixed generation error. So the purpose I want to do investigation that the research, they do research the peak, the current peak. The only method that collecting counting the egg mass in field and makes the decision the peaks. But I want to it more efficient. I just want to get some haplotype. If some haplotype very special to the univotum individual and the bivotum individual. So I found that several special haplotype very special to the univotum individuals. So we can use this haplotype to get some limited samples from in the field. We will know the population structure, so we don't have cost so much labor to do investment field to decrease the cost for biological control. I also do the whole genome sequencing and screen more than 200 SMP loci. So I also do the IBD analysis, isolated by distance. The mitochondrial gene have a genetic distance, very strong relationship with the geography distance, but the nucleotide SMP data have no relationship with the geography distance. Also to the cluster, it tells us the haplotype from the mitochondrial can separate incomparable to UM-Flint significantly. Still, the SMP loci still can separate the incomparable population to the univotum individual to the bivotum individual. So bivotum individual, some mixed error still can separate the bivotum individual and the individual. So this kind of SMP can be used in application for the monitoring. I also do some special genetic diversity and genetic structure analysis. We will notice that the SMP tells us the FST value within individual have a higher FST, or the among individuals have a higher FST too, comparing to the among regions and among locations. It tells us that in the location that the two kinds of voltinism individual have high gene flow. I also do some Bayes clustering. It tells us that in the mixed error, in univotum individual share a lot of gene with the bio individual. So it's proved that the high gene flow occurrence in the sympactual error. But I have shown some the figures about the many generation about the mixed error. They still come some characters of univotum characters, univotum patterns. It's kind of genetic answer, so continuous research will be done. Conclusion tell us that about the incomparable UM-Flint, the first time to kind of continuum is a hybrid individual in nature. It means that in the future, if you apply in field, so you should consider the host preference, because the similar species have two host preference. And about the univotism is that the SMP shows that some loci and some haplotype can be applied in field. So it will decrease the cost for monitoring. This is my lab. I have two assistant professors and one technician and several undergraduate students. So I have published papers about the two technicians. I got 3 invention patients, more than 8,000 hectares of the cornfield have applied my techniques, and I got some honors rewards. And I'm very thankful for USDA funding more research from America, and also have 6 non-principal investigative projects to support my research. I got 16 co-PI projects supporting this research. And also very like to cooperate with other people from the research center from France, And the USDA, always every other year I worked in USDA. When I worked in China, I still visit every other year, worked in USDA. I have some professor from Illinois. Every year I invite them go to China to do collaborations. But now I'm very interested in one kind of scientific question. This kind of basana should be safe, but sometimes it happened. It can make an invention in human being body. So it can reproduce themselves in people's liver and cause liver cancer. And also it cause skin burn, this kind of things. So I have the future work plan. I'm interested in that continue to study genetics and next generation develop new strains, because different target needs different new strains of the basana. There's a lot of the mating type gene. I'm 81-1, I'm 81-2, I'm 81-1-2, I'm 81-1-3. But so many, so I need some people who major in math to help me to moderate. And also need some people works in the computer science to use Python to organize the data like this. So I have to do a lot of research, basana in insect and in plant. Now I'm interested in do some experiment memo about the cancer. Also there kind of results. It's a virus from the fungi. So if I invest the virus to invest the kind of fungi, maybe it can cure this cancer potentially. I don't know. I need some research scientists who working on some cancer. I want to do some cooperation with them to do this kind of research. I have development of just only one software will help the benefit the population analysis, make the analysis very efficient. It's named the gene as based on the data of TBS data, but I want to continue to do research, this kind of research, and make a cooperation with computer science and develop new software. Thank you. So, are there any questions? Okay. Mm-hmm.
Pest control,
Mm-hmm.
would that maybe lower the risk of liver cancer? I don't know.
No, yeah, that's a good question. So usually the basana spread all over the world. When you eat all the plants, there's a lot of fungi in there, but need a kind of quantities. If need a quantity accumulation. So I remember that about 10 years ago, the research scientists have do some investigation about this. There's 8 species of the fungi to have established the same bond with all the plants. So when you eat everything, it left fungi spores. It should be safe, but sometimes if the spores accumulate enough quantity and just inoculation in the human body from the nose and the eyes, it can cause, maybe it can cause some liver cancer like this, but not very popular. But it's weird, but actually, when the temperature get over 30 Celsius degree, the spores will stop to development, to develop. But human being body is 37 Celsius degree. But usually on the different culture medium, the temperature should be safe, maybe increase, but very little people to do this investigation. But I think that most of the biological agent or the fungi is safe. They have experienced many times of the assessment. It should be safe. I don't know the reason why it causes cancer. Maybe special strains or just the patient gets too much of the spores. So I have got no report about this. So I need some people to help me to do some research on the cancer. Thank you. So are there any questions? So I just want to use traditional biological control method, use ecological method, use biotech, apply, use them to answer ecology question, but the techniques, use molecular techniques. So population genetics is a little from the traditional ecology. If somebody working on the ecology, I want to evolve their research, use my population genetic analysis techniques. Okay.
The results of the treatments on growth, both in below ground and years, I believe. How does that translate into crop yield?
It means that how to inoculate the suppression into the plant, right?
How does that impact the amount of agricultural products that is produced?
Okay. Yeah. Increase the products have two parts. That the first, to biological, because of the income model causes serious damage for the agricultural products. If I decrease the current infection of the income model, it can just not increase the products, but the only thing I can do is decrease the damage. So in American, the BT corn has been used very popular in the American, but in China, transgenic corn have not very popular because some organic food like sweet corn is BT corn is forbidden, that kind of things. So organic food no, so I cannot increase the products, but I can decrease the cost, decrease the damage. That's all. It can accelerate the growth of the plants, but I'm not sure I can increase the productive years significantly. I didn't do this kind of investigation, but it can at least can accelerate the growth of the stocks. Thank you.
Fungi is used as a pesticide in the States.
Yeah, actually, American the banana have been very popular. Use the crops in greenhouse. The different don't always use the big corn, but the sweet corn I have know this kind of data have been reported on website. But in China, use not only the fungi but also trichoderma. They also mix the two measures to control the pests in field to decrease the pest in sweet corn field. Yeah, but I found that if after I use it, I have a taste on this kind of years is have more sugar and more sweet than the control. Maybe accelerated the sugar accumulation. Maybe I didn't do this kind of the investigation, but tastes good. Trust me, it's safe. Usually it's safe, but there's only two articles reported that can hurt people. I don't know reason. Maybe special strains, or maybe they get too much spores from ears and mouth like this, and nose maybe. But I can do some use mouse to do this kind of investigation, like to do some investment, the safety about this. But I think that I have kind of enough virus to introduce it into the strains which have been inoculated in a human being. It can decrease the spores production in the liver. Maybe it can cure the cause liver cancer. Maybe I want to have a try. Okay. Any other questions? Okay. Thank you. Thank you so much.